Yeah, I'm going to talk about the role of epidemiology in uncovering novel or new, unknown, so far unidentified causes of disease. So some of you who've been involved in genetic epidemiology now quite a long time ago might remember this famous paper or discussion piece in Nature, which was the case of missing heritability. And this was because genome-wide association studies had just sort of started, with FTO in 2007 being the sort of the pin-up, most important of the early findings, for example. But what they were finding was that only a tiny proportion of the known heritability from twin studies, only a tiny proportion was being explained by identified variants from genome-wide association studies. And so there was much agonizing about this so-called missing heritability. Now, for traits like height, for example, which are highly heritable now, with massive sample sizes, with literally millions in the GWASs, together with whole genome sequencing, it turns out that for those sort of traits, there isn't very much missing heritability anymore. So the case of sort of missing heritability has certainly become less exciting. But we have the case of the missing environment, which hasn't attracted so much attention. Now, with genetics, with heritability, of course, we have a gold standard. We know what we should be getting. What we should be getting is something close to the twin heritability and be able to explain that variance with genetic data, with genotyped samples. Now, we don't really have the same, or it's thought we don't really have the same for the environment. But in fact, I think we do. And I shall, but first we've got to define what we mean by environment. And what I mean there is just things that could be modifiable. So modifiable causes that are not germline genetic causes of disease. Now, this isn't equivalent to all but germline genetic influences on disease, because some of the so-called environment, or so-called non-shared environment in quantitative genetics, is purely stochastic, i.e. it will be through stochastic processes that are not related to anything external. That's not of interest. Those processes that don't give us targets for prevention of disease. So the best approach, I still think the best approach to this problem, put formally, was by Richard Doll and Richard Peto in 1981, when they were charged by the US Congress to produce a report about the causes of cancer in the US. And what they pointed out, I mean, others had done, but they really put it together in a beautiful way. You know, they pointed out that we could know the in principle environmental contributors to disease by combining data on time trends. If disease is increasing or decreasing, and you have adequate ascertainment, then that can't be obviously germline genetic. The germline genetics doesn't change to lead to such increases or decreases. And there must be something tangible which is happening. You know, chance doesn't lead to increases or decreases. There must be some tangible factor which is leading to those time trends. The most powerful studies are migrant studies. When you look at where people have been born, when they move to other places, and then to see whether the risk of disease changes on migration, either in the first generation, actually in the people who move, or you see the change in the subsequent generation. So, you know, migrant studies as well are saying that something must be changing in the life experiences, which leads to change in disease risk. You know, geographical differences are largely unexplained by germline genetics, and geographical differences can be quite well indexed, or even in 1981 could be reasonably indexed by places where you had very good cancer registries. For example, and you had places with low and higher levels. And then finally, the environmental component could be known through identified causes, causes of disease that have been have unequivocally been proven, which in terms of cancer in 1981 was basically smoking. That was by far the major contributor of identified causes. So what they did is they looked at the best registration data in the US, registration and mortality data, and then they looked at places which had reliable data, which you think ascertainment would be reasonable, but low rates. And then they computed the proportion of disease that could be prevented if the people in the US had moved to the risk of the reliable, low-risk places, and said that that difference was preventable in principle, i.e. there must be some tangible factor that is generating those differences that could in principle be modified. So by environment, they mean everything. That includes the social environment, behavioural behaviours as well. It isn't just sort of external environment as it isn't just air pollution or something in the soil or pesticides or stuff. The environment are all those things that could be modified. And obviously behavioural factors were considered to be a major component of this. And by carrying out that exercise, they estimated that 75 to 80% of all the cases of cancer in the US were avoidable in principle. So this was quite an important finding, and generated much discussion, because it meant there was many deaths that could in principle be prevented, but only a fraction of those, were the cause known. And in 1981, that was mainly, as I say, smoking. Smoking made up the large component of what was an identified cause of cancer that could be avoided. Now, they made some guesstimates, and they were very, they put many caveats to these guesstimates. They made some guesstimates as to the contributors to all cancer deaths, and you see tobacco, the one that they are could be most certain about, contributing the third, which I mentioned. What's interesting, and actually I think was quite influential in epidemiology, was that they thought that diet was likely to be a major contributor. There was no real identified aspect of diet. This was the preventable in principle. They were saying that diet could contribute between 10 and 70% to the preventable in principle cancers, which is important. The other area was infection, where they just put a question mark at the end after 1% as a low to question mark at the high end. So these were their guesstimates, and as I say, these were influential. Many studies were set up explicitly to study the effects of diet. EPIC, for example, was planned following this, and in the setup papers for EPIC references Doll and Hill, Doll and Pito, and the large studies in Harvard, for example, on diet had a very major focus on diet. So diet was extensively investigated in the cancer field. And Richard Doll in 1977, in a review in Nature, excellent review, said, I can see no reason why the causes for the remaining common cancers should not be detected within 1 or 2 decades. So there was considerable optimism about that the epidemiological methods which had identified smoking as a pin-up. There was clearly this missing environment, and it was likely to be solved in 10 to 20 years. I mean, Richard Peter at the same time was writing something where he did say, the study of diet and chronic disease, all we know for certain is that we don't yet know very much for certain. So he was definitely saying this 10 to 70% estimate was guesstimate. And what really is a brilliant paper, and I suggest if people are interested in this area at all, it's a paper well worth reading. Was this exercise in what basically is a sort of triangulation of evidence that I was talking about on Friday. They put together evidence from these rather small human epidemiological studies on beta carotene intake. There was a sort of natural experiment in Brazil where some people were getting oil that contained a lot of high levels of beta carotene. There was animal experimentation. There was cellular studies, etc. And they're putting all the evidence together. They said that there was enough evidence to justify randomized trials of beta carotene. This is the only time in a Nature paper I've seen an editorial note added that cautioned unwary readers, if such there are, should not take the accompanying article as a sign that the consumption of large quantities of carrots or other major dietary sources of beta carotene is necessarily protective. So it's the only health warning I've ever seen in a Nature paper. And as discussed on Friday, of course, then the trials that did come forward were very disappointing. They certainly did not show material reductions in cancer risk, and there were some hints of increases possibly with beta carotene supplementation. So that was disappointing, and of course many others, as I talked about, then followed. Many other disappointing dietary intervention studies followed. Paul Brennan from IARC and I updated this. It was intended to be for 40 years, so 2021, but entirely because I'm very slow. It's my fault, not Paul's. It took us, we were a year late for the 40th celebration, but this is 41-year celebration of the Doll and Hill. Doll and Peter, sorry, their estimates. We did the same, but for the UK. We used I'm from the UK. Paul's from Ireland, sort of UK. He's born in the UK. We looked at the UK rates compared to the lowest reliable cancer registry rates, and we came up with this at 80%. The estimate of the preventable in principle is about 80%. Now, since 1981, I mean, despite Richard Doll's very considerable optimism, there have been sort of two major developments, really important in public health terms. The first being human papillomavirus, serotypes of that, and cervical cancer, which has led to vaccination programs which have been really substantially reducing the burden of cervical cancer, and H. pylori being identified as at least an important component of the risk for stomach cancer. And then a few other things, but certainly not a vast improvement in the identification, in filling in those known causes. In the UK context, the proportion of cancers for which a cause is known remains about a third. Now you think, well, the HPV, H. pylori, how come it hasn't gone up? The reason it hasn't gone up is because less people smoke. If you want to know what causes your cancer, it's very easy, just smoke. And then you will have a cause of then your cause will be identifiable. But as more and more people had become never smokers, the total percentage has not changed. There's only about a quarter of the cases in never smokers could be reliably attributed to a cause. And Richard Doll, in fact, wrote a piece 21 years after his things will be solved in 10 to 20 years, which basically said it had been very disappointing what had been identified. But as always, he says, while knowledge of the mechanism by which cancer is produced at the cellular level has increased dramatically. So that's sort of good, a bit of biological knowledge, but it's not great if you've got cancer because they haven't actually identified ways of preventing it, but they know more about how it's killing you. Paul, and our assessment last 2 years ago now was that that's not changed. So what might constitute this missing environment? Well, I mean, and now moving it not just from cancer, but considering disease in general, the same principles that Doll and Peter identified would apply to other conditions. It's just that cancer had more reliable data to allow one to look at differences in cancer. It's the same principles about time trends, migrant studies, et cetera, apply to other conditions. Well, I mean, for many causes of disease, it's very clear that adulthood isn't the important etiological period. I mean, the diseases, a lot of psychiatric conditions clearly developing from early life on, or maybe even prenatal life. In Alspach, for example, the polygenic score for schizophrenia predicts reliable differences of behaviours and reports by parents at age 7. I mean, not a single person has been diagnosed with schizophrenia by age 7, but if you use the genetic score and look at the kids at age 7, there were some very substantial differences between the kids with the high-end polygenic score and the low end. So that applies to many neurodevelopmental, neuropsychiatric conditions. They're observable early in life. And for other conditions, an important causal factor is acquired then, like H. pylori, fecally orally transmitted bug that's generally acquired in early life, relates to stomach cancer, peptic ulcer, and I'll come back about Epstein-Barr virus and multiple sclerosis later. In the cancer field, sort of a change, a real sort of paradigm shift in a way in the last 10 years is the fact that people started sequencing normal tissue as well as tumours. I mean, as you'd imagine, when sequencing became feasible at a large scale, focus was on sequencing tumours. But then when normal tissue was sequenced, and this really was a surprise. I know I was reading this and talking to people as these findings were coming out. A lot of people were surprised. Richard Peter wasn't surprised. He predicted this in 1984. But many people were surprised by this, and that you just sequenced normal colorectal cells, and many of them contained things which were driver mutations. They were things that, if they were in cancers, you would say that makes sense. This has the characteristics of a driver mutation, and there were large numbers of them in everyone. Endometrial tissue, then across all the human tissues that have been sequenced, and you're often finding these at early ages. I mean, you're finding, I think the youngest reasonable size samples for breast biopsy tissue is age 18, for example. Autopsy tissue has been measured, and the driver mutations are there. And indeed, in something which is one of the cancers, of course, for which there is rather little causal knowledge, for pancreatic cancers, it's not just the driver mutations. They're these pancreatic intraepithelial neoplasias, which are generally single clones, sometimes two, that are massive cells. In virtually everyone here in their pancreas will have several of these existing. And of course, for most people, for the large majority of people, they don't develop beyond being these remaining intraepithelial neoplasias. They don't actually develop into cancers in your lifetime and don't kill you, but they're there. So there's a sort of shift. There's been a sort of shift to think about not just mutation, as was always the focus of cancer, but actually something that changes in tissue that allows these driver mutation bearing cells, or these intraepithelial neoplasia, to escape from some sort of local control and expand rapidly. So that's really changed. I mean, you of course, it's just Darwinian evolution in the body, within the body. It's whether the cells with the driver mutations are out competing your other cells. It's quite simple process in that way, as John Cairns wrote about in the 1970s. Darwinian evolution is underlying this, so you've got to think of what are the selective processes that allow that selection to happen. So for several diseases, evidence points to early life acquisition an important cause, and classic epidemiological studies of cohort effects, ulcerative colitis, Crohn's disease, peptic ulcer, for example, possibly for early onset colon cancer, which I'll come back to, and also seen for which diseases which the cause is now known, such as peptic ulcer. This is a classically reprinted in the International Journal of Epidemiology some years ago, with a set of commentaries was Mervyn Susser and Zena Stein looking at the birth cohort effects, really dramatic birth cohort effects for peptic ulcer, which now would be attributed to H. pylori, which of course H. pylori medication treats peptic ulcer, and the H. pylori would generally be acquired in early life. Stomach cancer, to the extent the data are available, also shows cohort effects, and this was a review in 1986. Epidemiology run plan triumph. Epidemiologists couldn't claim any contribution to the fact that stomach cancer was going down. They had no idea why that was. Most of the focus was on dietary research, and it was a gastroenterologist who identified H. pylori and peptic ulcer, and then that became linked to stomach cancer. And there is a role for old school epidemiology. Here's some very old school epidemiology from that Dave Leon and I did many years ago, which was looking at the correlation of stomach cancer mortality in 1991 to 1993 with infant mortality 70 years before, for the median age of the deaths we were looking at. Now, of course, the children who died, they're not getting stomach cancer because they're dead. But if you were coming from a place with high infant mortality, you would be exposed to more of those factors which were generating mortality, which in 1921 to 1923 was largely gastrointestinal childhood diarrheal disease, infant and childhood diarrheal disease, for example, which would go with the sort of infections for which H. pylori would be one of those infections that would get transmitted in that way. And when you sort of looked at the top countries, and you thought, what factor, people studying diet, that was the main thing. And you think, what links Chile, Japan, and Russia? Well, the one thing is certainly not any dietary similarity one could think of. Russia was, of course, going through the period of very high alcohol mortality. Chile and Japan having the longest life expectancy then in the world. These are totally different places. The one thing they had in common was that those birth cohorts had experienced the very high rates, the conditions which led to high rates of mortality 70 years ago. So this is just I won't go on about this, but this is a plea in a way to say that there is a role for old school epidemiology. And one of the areas which is very disappointing in a way is, is how things like migrant studies, which are hard work, you have to go out and plan them and do them. There's been very, very few major migrant studies in the past 30 years. Most of them are still these are very old studies that we've done, and people don't do those sorts of studies very much anymore. For other conditions, life course Mendelian randomization suggests childhood is the important etiological time period. For those of you who can bear more, I'm going to talk about Mendelian randomization after. I'll probably skip this when I talk about it afterwards again. I won't go into this bit. But observationally, for example, your body size in childhood is a strong predictor of breast cancer incidence and mortality, both premenopausal and postmenopausal. This is work from Copenhagen by Jan Baker and colleagues, where they had school records where heights and weights were measured every year, and the BMI that was measured pre-pubertally. This is reported by Trajectory, but they did look. You could see at different ages. The higher BMI predicted quite considerably lower risk of developing breast cancer. And BMI is a phenotype for which you can do really quite good distinction using Mendelian randomization, and I'll show a lot of proof of principle on this and go into life course MR more in the next talk. But when performing multivariable Mendelian randomization, putting in both early life body size, BMI, and adult body size, you see a strong protective effect of larger body size. And looking at a whole host of potential mediators, the only mediator that sort of went in the right direction, i.e. childhood body size led to a lower level of it, and it increases breast cancer risk, is insulin like factor 1. It mediated a rather small component, but I mean, it's interesting that that's something, your insulin-like growth factor 1 levels at age 50 are influenced strongly by your body size pre-pubertally. And the other mediators went the other way, i.e. the larger childhood body size was related to higher bioavailable and free testosterone, which increases the risk of breast cancer, and it was related to earlier age of menarche. So what's happening here, when you see the things to the right, the mediation fraction is negative. The IGF-1 is a positive. So in fact, when you put in the overall mediation, actually makes the actual BMI protective effect of larger body size at age 10 stronger, larger, and there do appear to be two distinct, possibly distinct ecological pathways involving prepubertal body size and age of menarche, which in a way sort of repel each other rather than explain each other. And recently, Marina demonstrated that mammographic density does mediate a substantial component of the protective effect of childhood BMI. This is mammographic density at age 50. There's other conditions which one would think were mainly influenced, one would think were mainly influenced in adult life, factors that show strong effects of body size in prepubertally, prepubertal BMI. One is fractures in later life. So this is looking at fractures in UK Biobank occurring in 50, 60-year-olds. And childhood BMI is protective. If you look at the multivariable MR, it becomes quite substantially protective in the multivariable setting, and the higher adult body size is actually adverse, i.e. increases risk of fractures. So again, something in childhood which is setting the risk of even something like fractures, which would be thought of as a sort of adult life circumstances related condition. And the mediator in this case, again, really interesting to me, really interesting, is you've got bone mineral density in your mid-50s is influenced strongly by body size at age 10, i.e. a higher BMI at age 10 is related to higher bone mineral density at age 55, and the adult body size BMI effect is much smaller. So that's so there are these earlier life influences on these conditions which one would think of as being influenced by adulthood circumstances. So I mean, maybe one reason that this has been sort of maybe not inadequately thought about is that epidemiology has been driven, sort of post-Second World War epidemiology was basically epidemiology of coronary heart disease and lung cancer. But maybe, and then type 2 diabetes, maybe CHD, lung cancer, and type 2 diabetes are exceptions. They are the conditions that were indeed largely dependent on things happening in adulthood and things that were then reversible in adulthood. And this sort of brought the focus for the epidemiology in general onto that point in the life course. So for coronary heart disease, there's no influence of early life BMI, prepubertal BMI, once you take the fact that once you take adult BMI into account, the adult BMI is driving coronary heart disease. it's driving type 2 diabetes. Quite a nice proof of principle example. If you look at type 1 diabetes, the top two lines, two different GWASs of type 1 diabetes. In the univariate situation, both childhood and adulthood BMI relate to type 1 diabetes. Now, as the GWASs of type 1 diabetes were all children, they were all under 18 in the GWASs that were available, you know that the adulthood body size effects, BMI effects, can't be causal because you can't go, the opposite direction. Time's arrow can't point backwards. Your body size at 50 can't influence whether you develop type 1 diabetes at age 10. And this, so I think it's beautiful proof of principle data is that when you do just multivariable Mendelian randomization in two samples setting, your adulthood body size effect falls to none, which is as it should do, as it must do, because time's arrow can't go backwards. But this is suggesting that the sort of method is actually producing sensible results. Whereas type 2 diabetes is the reverse. Of course, the adulthood body size drives that, and there's no influence of childhood body size once you take adulthood body size into account. And fractionating the outcome phenotype further. Oh, it's not there. Anyway, I'll show it later. There's a fair amount of type 1 diabetes which occurs outside of childhood. This really is something that's been recognised, more recently. Andrew Hattersley being one of the people who's contributed to this. And in terms of public knowledge of this in the UK, the major contributor is our ex-Prime Minister, Theresa May, because Theresa May was a pin-up case of someone who had type 1 diabetes, but that wasn't diagnosed until she was in her 50s or 40s or 50s. Coronary heart disease, lung cancer, diabetes may be exceptions, and of course type 1 diabetes being very different. And in cardiovascular disease overall is a heterogeneous situation. So for hemorrhagic stroke risk, there is good evidence of childhood contribution. This is being the social class of one's father in a cohort which the father's social class was a strong influence on material well-being. You see a cohort in Scotland, you see a very large risk of coming from a household with a manual head of household, but for ischemic stroke, that was not seen. So this is with two adjustments. And another factor would be the number of siblings you have. Larger families obviously having less wherewithal, and also higher risks of infectious infant mortality and severe diarrheal disease in childhood. And again, the more siblings you had, the higher the risk of hemorrhagic stroke, but that's not seen for ischemic stroke. And the other condition that shows the same sibling association is stomach cancer. And also it shows the same childhood social class effects. It's childhood rather than current social class that influenced the risk of cancer of the body of the stomach. Deborah Lawler, as part of when doing her PhD, looked at stroke type, the crude distinction between ischemic and hemorrhagic stroke, and found every autopsy study in the UK she could find, and the ratio of hemorrhagic to ischemic strokes reported. And basically in the first half of the 20th century in the UK, the majority of strokes were hemorrhagic stroke, but then at around the time coronary heart disease was taking off, ischemic stroke began to increase and became by far the majority of strokes later, at the end of the 20th century. And then using those ratios, she applied them to overall stroke mortality and suggested that the estimated hemorrhagic stroke rate was just declining across the 20th century, whereas ischemic stroke showed an increase and then a fall. And you were missing the decline in hemorrhagic stroke because of the rise of ischemic stroke. because most studies didn't classify, couldn't separate the two. And in blood pressure was declining in the UK from at least from 1948 on, when a very large study of 18-year-olds from Glasgow University measured blood pressure. There was, of course, no one at 18 was on antihypertensives. Well, there weren't any antihypertensives basically in use then, but the blood pressure was declining quite markedly. And there are clear birth cohort effects in the decline in blood pressure in the US. And I'll skip my hypothesis about dehydration. I talked about it. I bored some people here about that already. So, and they might look into it. They might click it. So there's also childhood influences on atrial fibrillation and thoracic aortic aneurysm. UK Biobank, as well as being able to look at BMI at age 10. You could look at height at age 10, and you could do the same form of multivariable MR, and this is then the univariable MR, suggesting that greater height was associated with lower coronary artery disease risk, but higher risk of atrial fibrillation and thoracic aortic aneurysm. But then when you do the multivariable MR, it becomes adulthood height is protective against coronary artery disease and possibly peripheral artery disease. The childhood height becomes adverse, particularly for atrial fibrillation, thoracic aortic aneurysm. And since doing this, and this is now a med-archive publication, but we've also added the Million Vet study, which, as the name suggests, has a hell of a lot of cases, many more than UK Biobank, and the atrial fibrillation and thoracic aortic aneurysm effects become very markedly distinct. And there's taller childhood height is associated with greater atrial fibrillation and thoracic aortic aneurysm risk. 40 years later, your adulthood height is not related. It's not causally related. So yeah, so you know, in cardiovascular disease, there's a fair amount of heterogeneity. So as I say, maybe lung cancer, some other cancers, coronary heart disease, and type 2 diabetes are exceptions. And the other thing is, you know, that there's been RCTs of adulthood interventions. We've talked about, you know, dietary interventions, including the Women's Health Initiative. Yesterday, I talked about Friday, sorry. You know, dietary fat intake, which was thought to be a major risk factor for breast cancer. Interventions did not show that in the same way as none of the antioxidant vitamins, selenium for prostate cancer, etc. None of these were validated through randomized trials. Right the way through to things like the indoor air pollution reduction in lower middle income countries, in an attempt to reduce the very high rates of respiratory disease, especially in women. They have been very disappointing. This is to stop indoor cooking in Rajasthan, for example, and other places. And these trials haven't led to the reductions that was thought. You know, maybe things are happening earlier in life that mean it's not possible to reverse these by later interventions. So, you know, one aspect of these causes that maybe they're acting earlier in life than is generally thought, and maybe they're just more generally prevalent and not optimally investigated with individual level cohort studies. Talking about ubiquitous exposure, we can go back to Doll and Hill in 1950. One of the most remarkable statistics, and this is their case control study, the first very large-scale case control study of smoking and lung cancer. Of the 649 male lung cancer cases, only 2 were lifetime never smokers. Okay, well, that's in the cases, but in the controls, only 4% of men in the UK at that time were lifetime never smokers. You know, virtually every man had started smoking, and some had obviously given up. But you know, it's a pretty ubiquitous exposure. But most people were exposed. But you did have giving up with smoking, which is what actually generated the major differences in the dose-response relationship. And I think there's a lovely talk about ubiquitous exposures, there's a lovely thought experiment by Jeffrey Rose. And this is the, if you're going to ever read, if you've never read this, this is the best paper in epidemiology, in my view. The paper, it's only 7 pages long, and it's one of those papers every time you read it, you find, well, I find every time I read it, something I've missed the time before. And there's sort of in a one-line aside, Jeffrey Rose said, you know, consider studying lung cancer in populations where everyone smokes. So yeah, so let's do that. It's still a thought experiment. This is Bristol. Paul and Tom and Johan and Bjorn will at least will recognise sunny Bristol. It's always sunny. It never rains in Bristol. So imagine that the dictator came. I'm going to imagine that the dictator came in into Bristol and mandated that everyone had to smoke 20 cigarettes a day, every day. You got a packet of Capstan full strength, and before you went to bed, you had to prove that you had to show that you'd smoked 20 of them. You weren't allowed to go to bed until you had smoked 20. And you know, we're pretty wild in Bristol, so we'd go along with that, smoking, no problem. And then you imagine near Bristol is Bath, very posh place. Very posh. Those of you who've seen Jane Austen films of Jane Austen novels will think they've seen Bath. They're actually all, the indoor scenes are all filmed in Bristol, but that's it. If you look carefully, you'll see someone smoking inside. But and the dictator in Bath says no one could smoke at all, and everyone would comply in Bath, no problem at all. Everyone would not smoke. And then you follow these places up for 50 years, Bristol and Bath. And obviously Bristol would have more lung cancer. But within Bristol, remember, everyone is smoking exactly 20 cigarettes. So you've got your questionnaires and you're trying to study what's causing lung cancer in Bristol. Well, it's not smoking, everyone's exactly 20 cigarettes. You know, within Bath, within Bath, no one get lung, a few people get lung cancer, sorry, but no one smokes at all, so you couldn't identify smoking in Bath. And within Bristol, you know, why does one individual rather than another get lung cancer? You know, the first explanation would be people would say, oh, it's genetics. Well, you know, the heritability of lung cancer is only just a bit over 20%. And a chunk of that heritability is the heritability of smoking, the heaviness of smoking. So genetics would seem unlikely to be making a major contributor, say occupational exposures, etc. But maybe it's a bit rather more random. So between Bristol and Bath, the huge difference in the rate of lung cancer, nearly all cases of lung cancer, and the two together could be attributed to smoking. So at the population level, an exposure might be responsible for nearly all the cases, but within the population account for little of the difference in risk between individuals. So here's one of the people we might want to genotype to find out you know, how people live to be 100 as smokers. So Winnie is lighting a cigarette from the candles on her 100th birthday cake. She's been smoking for 93 years and she's not stopping now. So you say, well, you know, let's sequence Winnie. Let's find out let's ask Winnie how she managed to live so long. What did she do? One possibility is that it's just luck. You know, when she was 60, on her 60th birthday, postman comes to give Winnie her cards, still got cards in those days, knocks on the door, she opens the door, gust of wind, she just coughs up metaplastics. So she coughs, dislodges a metaplastic cell. If the postman had come at 10:59 or 11:01, Winnie would not be here. Winnie would be would have gone. But there would be someone, there would be John or Mary, there'd be someone who had been lucky, who would be there. You know, the number of people who would make it to 100 as smokers would stay the same. It would just be different people. It's just it might well be a sort of throw of the dice who gets there, and it might not be that rewarding studying individual when you've got ubiquitous exposure, studying who gets the disease and who doesn't. And then, in a way, almost more disturbingly, is then just thinking about within one individual. So you've got 2 kidneys. Hopefully, most people have 2 kidneys, and both of these kidneys will have the same germline DNA, identical DNA, obviously, at the germline. And if you eat all that healthy fruit and veg, if you don't, then whatever's in the healthy fruit and veg will be going to both kidneys. If you smoke, the effects of your smoking will be disseminated in the same way to both kidneys. If you drink a lot, the effects of alcohol will be disseminated to both kidneys. If you jog, the beneficial effects of jogging would be disseminated. If you were unfortunate enough to be anaesthetized with triolin, which was still used when I was a medical student for obstetric anesthesia, it's now a registered, it has been for quite some time, a registered carcinogen. If you were unlucky enough to be anaesthetized by triolin, then that would have gone to both kidneys. It's a renal cancer carcinogen. But what happens if you develop cancer of the kidney of one kidney, the other kidney has had exactly the same level of exposure to virtually every exposure that we could sensibly think of that we could intervene in. You know, not cosmic rays, but you know, can't study the cosmic rays, maybe a bit like the postman. What's your risk? What happens to the other kidney? Well, its risk of developing a primary, another primary in the other kidney, is elevated, but not massively elevated. The same as Julian Pito pointed out 25 years ago, and Richard Doll had actually informally pointed out in 1967. The same is true of breast cancer. If it develops on one side, you do have an increased risk of developing a contralateral tumor, but it's not massive. And that's when it's been matched on every single exposure. So maybe your kidneys are like the two people smoking 20 a day. When everyone's smoking 20 a day, that you know there's relatively there'll be some difference in risk, but a chance that will not be that easily identifiable. And you know, this is no surprise to people who work with clonal organisms. These are the marbled crayfish that are taking over Europe, starting from Germany. They're now spreading. Have they reached Norway? Is Norway full of marbled crayfish? They will be soon. You better get used to these fellas that come in for you. Anyway, they're parthenogenetic. They're clonal. They just split. So these are buds up. They're identical. Germline genetics are identical. Imagine you're a life course epidemiologist watching these crayfish. They're in the same aquarium. The environment's totally standardized. Trying to find out why one of them had got fat. It's unlikely you'll be able to detect the factors that lead to one clonal organism being different to the other, which you know leads to the Jeffrey Rosie's point, which is that you've got to look at population level exposures that lead to differences between populations to work out what you can intervene on. So what if the causes are ubiquitous within contiguous geographical regions? So you know within large. Often administratively joined regions, you know, the exposures, when exposed, the exposures are just spread. But so rather like smoking, but with smoking, you know, a simple questionnaire can find out about duration if you gave up, etc. Smoking was obviously, apparently Richard Pito told me that Richard Doll, like towards the end of his life, said we were very lucky to be studying smoking. You know, smoking and lung cancer was a lucky thing to be doing because it was this exposure that you could capture very well, simply. So you know what sort of factors are ubiquitous? Well, there are infectious agents which everyone gets. There's many infections which we all get, but the initial age of infection will differ. And maybe something as simple as when in your life course you develop the infection leads to major differences in disease risk. There's everywhere and forever chemicals, obviously, it's climate, water, including drinking water, indoor-outdoor air pollution, social stressors, anthropogenic light, which has been interesting with breast cancer, for example, obesogenic environment, mobile phone use, these things which you know within contiguous regions would be similar. So you know let's consider infections as one of these ubiquitous exposures. So a ubiquitous exposure if you had been living in parts of Africa and some of Cuba, Cuba quite a long time ago, Africa more recently, everyone got yellow fever. Every single person who was born there would get infected by yellow fever. If you got infected at age 2, there was a small increase in mortality risk, very small increase. If you were infected at 20, first infected at 20, there was a 30% mortality risk. Now, the reason why yellow fever became one of the first infections to be really seriously investigated by the US in Cuba in the late part of the 19th century, by the Brits and French and others in Africa, was because who do you think the 20-year-olds who were getting infected for the first time were? Oh, yeah, absolutely. It was the soldiers. They were being wiped out by yellow fever. That's why the US went to. That's why the US spent so much effort in yellow fever in Cuba. It was McKinley had invaded Cuba, and the result was very high mortality for the soldiers. So that's how that was detected. It's exactly the same infection, precisely the same infection. If you get it in infancy, small increased risk of death. But lifetime immunity. If you get it at age 20, very high risk. So maybe there's other infections, not so dramatic as yellow fever. There's some other infections, by the way, that are like yellow fever that really are dramatic. But maybe there'd be other infections. And Epstein-Barr virus would be an example of this. So massively oversimplifying a large literature would be that there's evidence that the disease risk for Epstein-Barr virus infection depends on the age of infection and potential cofactors. The EBV was discovered in Burkitt's lymphoma, so by Tony Epstein and Barr, who would send samples by Burkitt. They discovered this virus. And then it was said, well, this was a virus for Burkitt's, this was causing Burkitt's lymphoma. The only slight sort of knock on that was that they then discovered that virtually everyone had evidence. Once they could actually find out, they found out that virtually everyone in the places where Burkitt's lymphoma was prevalent had Epstein-Barr virus infection. And Burkitt's lymphoma is seen in regions, in places that have very early EBV infection. And there's evidence that very early EBV infection actually leads to greater activity. As you know, it's the virus stays with you forever, but greater activity, including seeing that in terms of antibody levels and fluctuations in antibodies. If you have very early EBV infection and there's malarious holoendemic, then the risk of Burkitt's lymphoma is high. There's evidence that's accumulating, it's still sort of accumulating, that other lymphomas and nasopharyngeal cancer, especially in places like in China and Thailand and possibly parts of Africa, are sorry? Is that okay? Okay, right. Of the nasopharyngeal cancer. It's EBV related, maybe a part of Hodgkin's and a component of non-Hodgkin's lymphoma might be EBV related. Then if you go without EBV infection, if you get EBV infection in a rich country where there isn't holoendemic malaria, and there are a few studies where people have actually seroconverted while they're under observation, in infants and young children, it's often asymptomatic. EBV infection at sort of age 14, 15 is related to a quite markedly increased risk of infectious mononucleosis, glandular fever, which is a post-viral syndrome. But it's a post-viral syndrome when you're actually never post-viral because the virus remains. And then if you get this later, and there's a paper that many of you have seen a couple of years ago in Science with the US conscripts, if you're EBV naive, and then you get infected in your 20s, there's a markedly increased risk of multiple sclerosis. So you have an infection, and if, like in UK Biobank, you test people, in a very high proportion of them, but it's still, the proportion still goes up from 40 to 60. A few people are still getting infected, do get partly random, partly non-random reasons. So you can have very different health outcomes for something which is, if you just treat the infection as a ubiquitous thing, it is ubiquitous. But the age of infection could be an important cofactor, and a condition where the age of infection really matters in terms of evidence of activity is with what used to be human herpes virus 8, and is now often referred to as Kaposi's sarcoma herpes virus. In Uganda, this is ubiquitous. This is all kids get infected with this, and they've got a lovely study now followed up to age 20. But where they show that they have the annual blood samples, and you could go back and say in which year interview did that child seroconvert, and the earlier you seroconvert, the higher your antibody levels are, and the supposition is that the higher your risk of developing Kaposi's sarcoma is. You need some sort of immunodeficiency to go with that. I mean, famously, of course, Kaposi's sarcoma, especially in richer countries, was only really outside of Israel and of other places observed with HIV. So, age of infection might be important, and one of a particular age that might be key, could be an intrauterine infection. And this is to oversimplify some quite lovely work with rodent models. Is that it can show that mice who are fetal mice, if they are subjected to an insult that increases IL-6 activity markedly, which you can of course experimentally do in those mice in sterile conditions, has very dramatically different effects on the brain development. Depending on exactly when in pregnancy it is. 2 days before that 3-day window, nothing. After that 3-day window, very little. If it's in that window, then there's a severe impact on brain development, etc. There's some evidence, but anyway, there's some human evidence on the same things. Now that's sort of a challenge. Epidemiologically, because how are you ever going to accurately enough identify exactly when infection or some insult occurs? So, maybe one reason for this for the fact that Richard Dole's suggestion that in 20 years we were going to identify most of the major modifiable causes of common cancers isn't realised is because the exposures we're looking for are ubiquitous. And what can be done? So large prospective epidemiological studies in similar populations starting in adulthood sort of worked for lung cancer and coronary heart disease and type 2 diabetes, but maybe it's not optimal for the risks that have not been uncovered in the last 40 years. Maybe more of the same on its own, more of the same isn't going to deliver. I think international studies that characterise normative data on age of infection by a wide range of ubiquitous infections would be really valuable. They're just, to my reading of the literature, these data just simply don't really exist. You don't have a baseline against which to study whether changes in that distribution of age of infection matter. We talk about ubiquitous exposures within geographically contiguous regions. So maybe those things that we were sort of told, we were taught, I was certainly taught, and I used to teach, were sort of rather low down in the sort of epidemiological hierarchy, like time trend studies, like ecological studies in different environments. Maybe those sorts of studies would be valuable, but using new technologies, environmental sampling, getting human tissues, including from autopsy studies, looking at mutation signatures, etc. Mutation signatures is quite an exciting, area in my view. So mutation signatures are when in the tumour you actually look for very particular motifs in the mutation profile, the motifs that might relate to different sets of exposures. And aristolic acid exposure is thought to underlie some kidney cancer in some regions at relatively high rates. And this is work by Albia from IARC, and she's going around these regions with really high rates in Romania, and she's going around slaughterhouses and getting, sheep liver and kidneys, etc. And they're looking for these mutational signatures that relate to aristolic acid exposure in these animal tissues, because the plants are in the grain fields. That's how it's thought this gets into the diet in some of these regions. So it's a really interestingly different sort of data collection than epidemiologists usually think about. But so collecting these sorts of data may be valuable. These are two sort of pin-up mutation signatures. One, the SBS4, it's called, is a smoking signature. Another one is a UV light exposure signature. So it's tobacco, UV light. There's aflatoxin, which relates to liver cancer, especially if combined with hepatitis B infection, relates to very high rates of liver cancer. Aristolic acid and renal cancer, as I discussed. And then there's some endogenous signatures. And in a recent, published a couple of weeks ago, a lovely paper, highly recommended paper from Musicraft's study. In Japan, they discovered the signature, which only exists and basically didn't exist beyond above 2% in the cases in all the rest of the world. But in Japan, was seen in 72% of the cases. And so the supposition is that there is some exposure which is unique, which is only seen in Japan, which is might be generating an important proportion of the renal tumours in Japan. Now, when you can then treat this, you can treat the signature as something that you study you look at to try and find out what is that exposure which leads to this signature. That's doing the similar sorts of work as has been done in Romania, including actually looking at migrated cases that migrated into Japan, etc., to try and look at the determination of this signature. So this is the sort of technology, obviously, it's relatively recent technology. There's interest in a supposed global epidemic of colon cancer in the under 50s, which supposedly shows birth cohort effects. And there's a signature of SBS 88, which is induced by a genotoxic E. coli, which might be contributing to this. And there's a lot of extensive interest in working through this. Although on the there's a recent cold shower from Norway, from the Norwegian Cancer Registry came out, because everyone's reporting these increased incidences. You notice Norway does have a remarkably increased incidence, but the mortality isn't increasing. Now, this could be because early onset colon cancer has always actually had slightly better, if it's not related to a major mutation, has a better major cancer mutation, cancer family syndrome, has better survival. But there's definitely some interest in what might have happened that led to this. Other sources of data that could be really valuable about exposures, but also as mechanism, would be tissue-specific DNA methylation data, which, for example, in the autopsy studies, but in studies with tumour and normal tissue, you can use to look for environmental exposures using DNA methylation data. We know that blood DNA methylation data, for example, gives you a very good indicator of smoking and arsenic exposure and a few other exposures. But blood is probably the wrong tissue for most most conditions. So if you get this really nice couple of recent papers using animal models and some human data, if you get the specific data, you might be able to notice these epigenetic signatures which relate to a particular exposure, but they might also be part of the mechanism. They could be, this could be part of the mechanism which makes that tissue a fertile bed for positive selection for the pancreatic intraepithelial neoplasms or cells with driver mutations. So, so getting tissue might seem a bit morbid, but collecting such tissues might be a way into identifying exposures. Obviously, need to work with environmental scientists. This is a nice, I like this because it's near Bristol in Tube Magna, from above and below the part where I don't know, I'm sure no one follows the UK news, but one of the major news stories over the last few years is that the water companies have just been dumping sewage unbeknownst to people into rivers. And this was a study of looking at using mass spectrometry and there's just thousands of things in the water downstream of where this was being added that weren't in the water, but above where it's added. An interesting thing which was suggested in the sort of 1940s, 1950s, or 1960s even, that because wild animals weren't developing cancer then, it wasn't environmental in the old ways we think about environment. But there's there's more recent evidence that oncogenic processes are becoming commoner in wild animals. So these might be the sort of canaries in the coal mine in one way, but also there might be ways of identifying exposures through, for example, mutation signatures that are being done on slaughterhouse meat, but also through looking at accumulation of exposures in the way that fish, for example, concentrate certain exposures in water and are better at concentrating those things than maybe some other forms of chemistry. And of course, obviously, Mendelian randomization could play in here, because Mendelian randomization can tell you whether something is likely cause, even when everyone's exposed to exactly the same level. And I mentioned that smoking, heaviness of smoking, there's a genetic contribution to heaviness of smoking. There's one variant, of course, which is much stronger than any other variant. It was by far the top variant in the smoking and the lung cancer GWASs, which was a nicotine receptor variant, CHRNA 3/5, which leads to people smoking more heavily. And we showed a long time ago that people who smoked exactly the same number of cigarettes, if they carried the high risk, or the homozygous for the high risk variant, had 30% higher cotinine. I mean, they were probably puffing them harder. So the dictator isn't able to stop you, not smoking quite so far down the butt and not being quite so enthusiastic in inhaling. They're not going to be able to do that. And so, and if you look at people who smoke exactly the same number of cigarettes, who report the same number of cigarettes, the Chrona5 variant relates to higher lung cancer in people who smoke the same number of cigarettes, because we showed that in this paper. We showed it with some data, including data from Norway. It's because they are smoking more heavily. They are getting more exposed. Even though they're smoking 20 cigarettes a day, they're getting more exposed to the toxins in smoking. So if you have in Bristol, you genotype the people in Bristol, you'd find that the people who were dying, who were developing lung cancer, would carry these high-risk variants more. And that would tell you, or point you very strongly towards smoking as being the exposure. And possibly my favourite recent example of this is genetic variation related to malaria risk and Burkitt's lymphoma. Of course, you can do Mendelian randomization. This is a terrible photograph because you can see me holding my phone pointing at there. But I was in Uganda in the MRC, what used to be the Virus Research Institute and MRC unit. This is Burkitt's original map of when he was identifying cases of Burkitt's lymphoma. And he pointed out these were, you'd see it only in holoendemic regions, or holoendemic for falciparum and malaria. Is of course you've got sickle cell trait, which is a very strong proxy for your risk of developing falciparum infection. And sure enough, sickle cell trait, the heterozygosity protects quite strongly against Burkitt's lymphoma. Many this has been replicated, etc. The thing I was most interested in, because some people here know I'm very interested in history, is this had been first thought of in had been thought of by Malcolm Pike, very young Malcolm Pike, and then in 1970, although his sample size was too low really to establish it. But you can use genetics, germline genetics can be ways of identifying ubiquitous exposures when everyone is exposed. So there's almost tangible, but as yet unidentifiable, modifiable causes of disease, the weight discovery. I won't say you've got to do it in the next 10 to 20 years with this, all these great methods, but certainly larger and larger cohorts of the wealthy, middle-aged. I'm a participant in UK Biobank, so I'm allowed to say that 5% response rate. I'm unlikely to crack the problem, so epidemiologists should think of other things than just getting very large cohorts. This is I would like this. The policeman finds the drunk falling along and says, "Is this where you lost your wallet?" And he said, "No, I lost it at the park, but this is where the light is." Is that maybe, UK Biobank is available, you can get papers out of it very easily. So you just crank the handle on UK Biobank, and maybe it's not going to crack the problem. And these are people who've really helped with some of this. Ada's person who's doing this heroic data collection exercises, who's running those. And for further reading, this is what you want. It is a masterpiece. It's beautifully written. It's the best introduction to epidemiology there is. I gave a Cutter talk at Harvard, and I suggested people read it, and I put this up, and as soon as I'd given it, my phone rang, and Richard Peter rang me up and said, "Thanks so much for the advert." So then I was at a meeting in Barcelona recently that Richard was also speaking at. So I looked again. It's £1.53. I said this is the best value you can get in epidemiology. It now costs £132. It's the cheapest one you can get. So I told Richard he owed me a few pints. I put up the value of his book dramatically, but it's a brilliant book. And I mentioned the fact that Richard was one of the people who wouldn't have been surprised by the mutations. There's an absolutely beautiful chapter he wrote in 1984. Which is called On the Need for Ignorance in Cancer Research. And we recently reprinted it in the European Journal of Epidemiology, so you can read. It's a wonderful piece, honestly. It's very short. And then with Paul Brennan and Bert Hoffman, we wrote a commentary on it, and Alan Balmain wrote one about promoters in cancer. So that's reading. That's it. Thank you.
Thanks a lot, Paul, for a great talk. Really encouraging us to rethink our conducting in the field. Questions or comments for Jones?
Really just comment, if anything. I mean, if you remove all these risk factors, such as obesity, which is taken over from smoking, smoking, alcohol, you name it, you will still have more cancer cases in 2040 than you have today.
Is that true? Well, because of survival.
Exactly. Because of the immune surveillance system.
Yeah.
So one single factor that's playing with us here. So that's a challenge for this. I mean, so the 1/3 is probably going down these days. Maybe. You understand what I'm saying? That
of types of incidents.
Yeah, I assume that 1/3 or a little more is environmental, or we have only discovered the stochastic, or certainly the mutations will increase with age.
Yeah.
And of course, environment will play a role, but you know,
Yeah.
there will be more stochastic mutations with age, probably.
Yeah, what an interesting thing I think, which is sort of studyable, is whether the mutations at old age really matter. Is whether whether the possibility, the Darwinian struggle, if you like, the length of time matters, and this is, I mean, as you know, Richard, the two Richards and others wrote a lot about, you know, about with smoking, about the basically, you know, it's just starting early, and the time, and maybe the mutations that are, the older age mutations that are occurring don't have the same probability of actually developing into tumours. I mean, if you look at, for example, because it is simply Darwinian, you can use the methods that are used for identifying the earliest common ancestor, that you can use for identifying early common ancestor in human population, in animal populations, populations of life. You can use those methods for dating the emergence in tumours, and a very detailed analysis of kidney tumours, for example. Age 10 was the age that was the sort of average median sort of age of when they think the mutations, the driver mutations occur. So maybe the driver mutations occurring at age 60, they don't have the time to, they don't have the time to evolve and take off. That's a speculation. We don't know if it's true, but it's possible.
I mean, one thing you have to consider is clonality. You are one clone until you turn 55.
Yeah.
Then you suddenly have multiple clones in yourself because you immunologically cannot control the clonality issue. That really boosts the incidence of cancer dramatically.
I mean, that's an interesting point. Whether that's obviously with clonal hematopoiesis, for example, has been used a lot for sort of, you know, you do see that taking off in that sort of way. Maybe though for the mutations in the other tissues, there's not the same take up. It depends on the cell division rates, for example. I mean, it won't be the case for brain, obviously. So yeah, no, I mean, it's definitely one of the most interesting areas, I think, is actually trying to delineate these trajectories. So I certainly think what you say, might be important, might be true. But it's all, yeah, in some circumstances, like clonal hematopoiesis, you can sort of demonstrate it. But maybe for solid tissues, it's not so much. Maybe for solid tissues, one's actually looking at modifiable promoters. It's the things which influence the tissue. But people have pointed out that what's a promoter for something which is, your pancreatic intraepithelial neoplasm might be a distant mutation which then changes the tissue microenvironment. Is being changed by those things. I mean, certainly being changed by, I think the evidence for stable epigenetic changes, change in the tissue microenvironment is growing and is strong. So, but I mean, yeah, studying what actually influences the microenvironment the tissues is key. I mean, I think the bottom, the sort of the headline notion that there must be environmental factors we've not identified is the case for, I mean, there's clinical conditions like pancreatic cancer, a big chunk of non-Hodgkin's lymphoma, chunks of many cancers. There must be something that in principle could be identified. But I think some of these things will be so might be so extremely difficult to identify, especially if you basically got the situation like with Burkitt's, where you've got two ubiquitous exposures. You've got early Epstein-Barr virus, and then you've got malaria, and it's the two coming together at a particular age, which influences risk. But there that was identified. So, but maybe because it's an infection and it's easy to do infections and possible to study it that way.
You introduced the risk factors work in a temporal way and in the ubiquitous way. I guess a lot of diseases are multifactorial, and there have already been many studies like to study the interaction of genetic environmental factors.
Yeah.
Even though we have a pretty large force nowadays, but considering the rare disease, most of the disease of interest are rare, and could be the power is an issue that we don't identify new risk factors. And also my question is, are there any methods to investigate the interaction of multiple environmental risk factors and can still work around the power issue?
Yeah, so I mean, the Burkitt's is a great example of interaction of two factors, two environmental factors: Epstein-Barr virus infection early on, and then malaria. Yeah, so certainly it might be the case that these ubiquitous, what you're looking at are these overlapping multiple ubiquitous exposures, and then delineating those interactions will be difficult. I mean, the sort of goal of epidemiology and public health are to reduce population risk. Is what we should say: is there some fundamental factor that, if not present, whatever the other factors are, drives risk? And then, for lung cancer, we know there's smoking. For cancer of the body of the stomach, taking out cardiac cancer, H. pylori infection seems very important, but there could be a second factor that might be required, and that might be why the rates are rather low in some African countries where H. pylori infection, early H. pylori infection occurs. So and then you would want to use the sort of methods like mutation signatures. I mean, it's sadly there's been rather little done in stomach cancer in that regard. But these technologies these techniques are getting, the cost is it's the reverse Moore's law. Well, it's like Moore's law in terms of the more sequencing you can get for the same amount of money. The cost of doing these things is of course plummeting, and so there's real possibilities of generating these on tens of thousands rather than the few hundreds that we currently have. So yes, that's right. So I do think I think the mutation signatures, tissue-specific methylation data, etc., are really quite promising ways of trying to identify these missing environmental exposures.
Yeah, thanks again, George, for a very nice presentation. You have a fantastic ability to synthesize, almost like a historian. But you were saying that you were having some perhaps worrisome thoughts about this large cohort studies, and for us who are planning some 5 study.
Yeah.
I guess you have been discussing this also, but could you share some thoughts on what we should do when planning this next Hunt 5 study?
Yeah, I mean, I think you're a contiguous area, so these might be things which are ubiquitous in the area. But we talked about, for example, looking at historical data on there was different levels of economic, when infant mortality fell quite differently in different areas. I think, and I think just environmental sampling. That's not. I mean, I'm always sort of surprised that people don't just get people to put tap water, whatever they're drinking in the house, just take a test tube full of tap water people are drinking, because I mean, certainly in some places I'm working, obviously I don't know about that. Maybe everyone gets absolutely the same water here, but in a lot of places that historically hasn't been the case. But just thinking about things you could get that would index environment, it would be good. And I mean, I'd actually plan to consent people for collecting samples after death. Though I mean it's obviously controversial whether one should do that. I mean, in the Caerphilly study that I was involved in 40 years ago, we have got a large number of brains. People just consented to have their brains taken when they're dead. And we've got many hundreds. I think maybe 1,000 out of a sample of only about 2,500 originally. So thinking about consenting people for that, I think would be amazing. I mean, as I say, maybe you think the response rate might go down if people think, but I'm not sure we would. I think if people, I think getting something, it interests people, getting something which isn't, in the Caerphilly study, of course, these were guys were 100% of them smoked and drank, they were all men, were minors. Filling out questionnaires about their smoking and drinking, they were not so interested in, but talking to them about dementia and getting their brains more interested. So you might find you get interest, you actually increase the interest if you start thinking of those things. I mean, one thing we actually tried in a study in Wales when I was there, and what didn't turn out to be as interesting because, as I hoped, was just to ask people what they think caused disease, what they think, why are there? I went to study right the way across Wales. Why were they in a particularly high risk of silicosis area? Well, the obvious answers to that. Why? What people think caused things. As a way of also engaging people, also as a way of sort of hypothesis generation. But the bottom line is you've probably got is that I think tissue collection is I think that's the tissue collection plus some form of environmental sampling.
Thanks, Josh. You really challenged us in how we should conduct the data collection, how we should really design our studies to identify some of these still unknown causes. I think we'll have a 15-minute break now. We haven't made, so for those of you who are now joining online, we haven't made a specific Zoom link for the next session, which is a workshop, but really.
No, it's just a talk. I explained that I can't actually do anything, you see. So workshop suggests I'd actually be showing you how to do some analyses. Well, I'd sort of have one of those hand-driven calculators. I'd be So but I asked people to email me. No one. I wanted some, horrible questions about Mendelian randomization, but no one dropped me any emails about. So it's about that being about Mendelian randomization because I was happy to talk about anything, but it was just be chat, and I'm just very interested to hear people's experiences and maybe to talk through some of those issues. But otherwise, it'll just be me. It'll be me talking. And then there's beer, I think.
Yeah, and we'll keep the Zoom link open. So if any of you online want to join also the next session, just please stay on the Zoom link. Yeah. Thanks again, Josh. Thanks a lot for listening.